Can you hear me well? Okay, so today I'm gonna start to talk about coevolution in heterogeneous environments and how it actually does not affect coexistence between two competitor species. And when we have species that are competing in the same environment, the presence of multiple types of resources can actually change how they coexist, because the existence of multiple resources can lead to the evolution of character displacement, where each species will explore different resources, so then reducing competition. And this can, of course, change the way that species coexist. And previous studies have shown that by having different types of environments changes the coexistence patterns between species. However, we still don't know how the presence of heterogeneous environments actually changes the coexistence between species. And we can actually do this type of prediction by using modern coexistence theory. In this framework, we can predict if species can coexist or not by determining the niche, which is the x-axis in this plot, and the fitness differences. The niche differences basically dictate are defined by the strength of the inter versus interspecific competitive ability between the two species, while the fitness differences are determined by the growth rate and how well the species are able to explore a resource. And in this type of plot, coexistence will happen whenever fitness differences are lower than niche differences. And previous studies have shown that that evolution of one species can actually change the way the competitor responds to that species. At the same time, other studies have shown that co-evolving with competitors may change or not the coexistence outcomes. However, we still don't know if evolving in the presence of a competitor versus co-evolving with that competitor can actually change coexistence outcomes. So basically here we have two main questions. Does co-evolving or evolving with a competitor in a heterogeneous environments change the intrinsic growth rate and the competitive ability of the different species? And the second one is whether coevolution in an heterogeneous environment shape coexistence outcomes. And in order to answer these questions, we're using spider mites, which are agricultural pests. And we're using two species, Tetranicus evansi, which is a tomato specialist, and Tetranicus urtica, which is a generalist. And although evansi usually outcompetes urtica on tomato plants, in the field we actually see them coexisting in the same plant. At the same time, we've also seen that tomato plants are hyperaccumulator of heavy metals, and both species are actually negatively affected by cadmium. In presence of high cadmium concentrations, both fecundity and offspring production in both species is reduced, equalizing fitness differences between them. Using this setup, basically what we did was then create an experimental evolution setup in heterogeneous environments where spider mites were actually exposed to plants without cadmium and with cadmium at the same time, and were maintained through several generations. We had for each species a control selection regime where the single species was evolving in a heterogeneous environment. We had a selection regime where there was one species that evolved and was transferred each generation, but it was always competed against the same competitor. And then I will refer to this regime as the ancestral competitor regime. And then we had the coevolution regime, where both species were transferred each generation, and so they were co-evolving together. And I will refer to this as the co-evolved competitor regime. For each of the selection regimes, we had 5 replicate populations that evolved for 35 generations, and for each assay that we did 2 generations of common garden to remove potential maternal effects and equalize the environment across the different selection regimes. We did 2 different assays in generation 15 and 35 to quantify changes in the competitive ability. And basically we had 3 different treatments. One, where we put one female and we measured how much offspring it produced after 1 generation. Then we had one focal female exposed to a different gradient of conspecific competitors to measure interspecific competition. And then we had one treatment where we had a focal female exposed to a different number of heterospecific competitors to measure interspecific competition. We then took the offspring production of all of those females, and we fitted the Riker model to estimate the intrinsic growth rate, the interspecific competition, and interspecific competition. And we did this both in the cadmium and the non-cadmium environment. And so now I'm going to give you some results. First, I focus on the intrinsic growth rate. And here you'll have always the same type of plot. In the x-axis, you will have the selection regimes. We have the ancestral competitor, the co-evolved competitor Forte urtica and Forte evansi. in the bottom part, we have the no cadmium selection regime, and the upper part, the no cadmium environment, and the upper part, we have the cadmium environment. We have generation 15 on light blue and 35 in darker blue. And because we're using the single species regime as a control, basically, whenever the values are above the line, it means that the regime was showing higher changes than the control regime on that specific trait, and if it was below, less changes than negative changes in relation to the control regime. So starting up, we first see that for the no cadmium environment, coevolution or evolution with a competitor actually increased the intrinsic growth rate, but it was for the better competitor, T. evansi. While in the no cadmium environment, we actually see that there are no particular changes observed for any of the species and selection regimes. Looking at the interspecific competition, so the response to conspecific competitors, we actually see that, again, coevolution or evolution with a competitor actually increased the interspecific competition for the better competitor. And in the cadmium environment, it actually led to coevolution initially led to a decrease in interspecific competition, but this pattern was lost at generation 35. Then, for interspecific competition, we did the test with two different types of competitors. One was the ancestral competitor, the one that was evolving with always one of the species, and then we also measured against the co-evolved competitor. So, in relation to the ancestral competitor, basically what we saw is that, again, for the T. evansi, so the better competitor, coevolution increased interspecific competition, but only in generation 35. And this also happened in both the non-cadmium and the cadmium environment. Looking at the co-evolved competitor, now our line becomes something different. So we're comparing, the control here is the response to the ancestral genotype. So we're going to see how the fact that the competitor evolved affected or not the sensitivity to competition of our focal species. And so if it's higher than the control, we're seeing that the focal species is more sensitive to the effect of the competitor. And what we see here is that when evansi co-evolves, then it is able to affect more the Urtica focal species, okay, in the no cadmium environment. However, this did not happen in the cadmium environment. Now, the question is, do these changes that I've showed you affect the long-term coexistence patterns of the species? And to do this, basically, we're using the structural stability approach that allows to predict coexistence, including several types of interactions. And this is important because we've seen facilitation in our system, and this means that this is not directly integrated into the modern coexistence theory framework. And here in the structural stability, basically we have in the x and y axis, we have the intrinsic growth rates or the growth rates of both species. And inside of this matrix of intrinsic growth species, we can have a region where they can actually coexist. And this region is delimited by the strength of intra versus interspecific competition in the species. And it's called the feasibility cone. If our growth rates are inside of this feasibility cone, then species are able to coexist. Otherwise, one of them will be excluded. And so here we're going to see two plots that are for the non-cadmium and the cadmium environments. And here we have generation 15 on light blue and 35 on red. And what you see is that both in the non-cadmium and the cadmium environment, actually most of the dots are actually in the exclusion region, which basically means that there is no changes in the predicted coexistence outcomes between the two species, although there is some changes and some increase in niche differences, especially in the no cadmium selection regime. But in the end, coevolution did not affect the predicted the coexistence outcomes in any of the environments. So, to summarize, basically we saw that evolution or coevolution in the presence of the competitor increased the intrinsic growth rate, mostly in the no cadmium environment, but only for the better competitor. And this led to an increase in the interspecific competition for exactly that species, and also some changes in the interspecific competition. Nevertheless, this did not affect the predicted coexistence outcome between our species. And this was mostly caused by the fact that although we had an increase in niche differences in some of the environments, there was still very high fitness differences between the two species, which leads to the exclusion of the worst competitor. So in the end, basically, we saw that coevolution does not affect coexistence patterns in spider mites. And I would like to thank the Spider Mind Squad, the funding, and you for your attention. Happy to take any questions. Cool, I think.
Excuse me.
Yeah.
I didn't catch. How did you define or distinguish or quantify better competitor versus worse competitor?
Basically, so the way that we define better or worse competitor is related with how well they perform in the tomato plants, and also previous studies that we've shown that when they are competing in a tomato plant without anything, the Evanzi usually excludes Urtica after 2 or 3 generations. So this is information from previous studies. Okay? So basically.
So during the evolution experiment, did you control or do anything with densities of the?
Yeah, so we had each generation, always 220 females were transferred for each selection regime to maintain control densities.
One of your treatments was with the cadmium, so as I understand it, it has a negative impact on both of them, so you diminish their interaction in a way. So do you have any expectation if you did an opposite treatment, so like with a plant that is more nutritional or something like that, that they would be happier? Do you think this would increase the impact of the better competitor somehow?
Well, the thing is that, so here what we see is that actually, because the cadmium is such a poor resource for them, they actually focus on exploring mostly the better resource. So if I would take this and do it for an even better resource, I would say that the better competitor would even explore much better that resource and would exclude much faster the worst competitor, Urtica. Okay.
Okay, I'm not sure this makes sense, but you predict that there should be no long-term coexistence, right?
Mm-hmm.
And yet it evolved for 35 generations or so. So how do you?
Because we maintain the density each generation, right? So what happens is you transfer to 120 females, and then after 1 generation you transfer again to 120 females, even though the better competitor has much higher density than the lower competitor. So they are able to get at least one out, but if you maintain this through more time, and we actually have an experiment that I didn't show here, but we have it that, for example, for 2 generations, then the ratio between the species becomes much different. So basically, Evanzi will take over like 95% of the population. So it's very quick. The problem was that we were maintaining them artificially each generation. So we were directly impacting in the dynamics of the system.
So you said that these two were coexisting in the wild, but they don't coexist in the lab. How does that come?
Well, one of the things can be also, so another study that we've done before, it shows that when the fourth competitor arrives first, it actually is able to maintain coexistence between the 2 species. So by just arriving 48 hours before the better competitor, it basically does niche preemption and occupies the niche that the better competitor prefers and allows to maintain coexistence. And this is in the lab. So we know that this type of priority effects can change the dynamics. So it could be one way of them to be able to coexist in nature. Any more questions? No? Then going.
Okay, good morning, everybody. I was just checking, it was 9:45 on the dot. Am I okay going ahead? I've not done this before without the chair of the session, so very good. So hello, my name is Graziella Iossa. I am from the University of Lincoln, and I am going to talk to you about a study which has just been published on the Royal Society Procreosoc about coevolution between eggs and sperm in insects. So, what type of selective forces operate on gametes? We kind of know a lot about the selection forces that operate on sperm, and you have obviously an obvious form is sperm competition. So in this little schematic, you can see kind of an outline of an egg. Bear with my not very good drawing skills, but I hope they make sense. And obviously the fertilization environment within the female is really important. And we know that, for example, the reproductive tract has a strong effect on the morphology of sperm. So what about the eggs? I think the eggs are not as well studied. Eggs have a different type of challenge in that they, first of all, need to be fertilized within the female tract, and therefore there's an internal environment within the female body, and then they need to be laid out in the external environment and also survive so that the embryo can make it, essentially. So the forces that act on eggs are dual, and also have the environmental effects are also very important, and so the kind of protective challenges that the female have are different. So I think I didn't say very well this, but the forces that are at play are different for male and female traits, essentially. And we have, of course, also, in addition, pre- and post-copulatory pressures and pre- and post-laying pressures. So it's quite a kind of complex environment. Okay, so I'm sure you probably all know that on the left-hand side you can see what a normal egg for a vertebrate looks like. So that is permeable everywhere. So anywhere where the sperm enters, it can fertilize the egg. But insects are different, and you can see an insect egg on the right-hand side. And around the egg, there's an eggshell, which is not permeable, and it's the chorion. And therefore, the only point at which a sperm can enter is a micropyle, which is this trait which insects have. Micropiles have spurred my interest for a few years now, and not just because eggs are beautiful, but also because this trait is extremely variable, varies within species, between species, and within the same female. So there's a lot of unexplored variation there. And I'd like to argue that we place a lot of importance and interest on sperm differences across insects, but we don't place enough importance to the egg. And if we want to understand sperm and egg, of course, we need to look at both traits because they co-evolve. They need to interact during fertilization, therefore, they must evolve. So, I want to show you just a little bit of this variation. Here you have a butterfly egg, a green-veined white, Pieris napae, and this is a scanning electron micrograph of one. So you can see the full view of the egg on the left-hand side. Typically, but not in all insect species, micropiles, this place of entry for the sperm is located in the apex on the top side of the egg. So here, what you see at the bottom is actually a leaf. And if we zoom in into that tip, we can see there is this micropilar plate, which is basically like a landing pad for the sperm. And the pore where a sperm enter is the actual micropyle. So we zoom in even further, and we can see in the bottom right-hand side about 5 micropyle if you squint. But I've put some arrows for you to kind of point them out. Micropiles are hugely diverse. As I mentioned, I have been looking at them for a few years now. In my previous work, I have shown that across insects, they can vary from as little, which is probably the ancestral form of one micropyle, to 120 in Hymenoptera. Not all of the micropiles are functional, so not all of the pores are open, but they are really diverse, and I think they're super cool. We should study more of them. So on the left-hand side, you can see what a kind of more primitive insect, the firebat, looks like. That's the micropyle in that case. You can see it's a very large entry point, one only, and away you go. But on the right-hand side, you can actually see Drosophila species, and you can see that, so the kind of, I don't know why I don't have a pointer. Can you see my cursor? No, good. So the black image is, you're probably all familiar with the Drosophila eggs. These very long appendages are just breathing appendages, but in between, in the middle, very small, is that micropyle. And you can see that there is a canal within that. I hope you can squint and see. So that tube that protrudes out of the egg, that's the micropyle. So it can be going inwards or going outwards. This is part of that variation I was telling you about. Sperm and micropyle interact. This is a fantastic image, and it shows the canal and how the sperm enters the pore and the canal in the cricket. And therefore, we looked at morphology of sperm and egg, and in an evolutionary and phylogenetic context across insects. So what did we find? We found on the left-hand side, you can see that we found that size across sperm and egg evolves at different rates, and sperm appears to evolve faster, 3 to 4 times faster than egg traits. So in this case, we're looking at sperm length and egg and length and width. So the traits are evolving at different rates. And in again, if we dig down into the orders, so you can see on the right-hand side, this is again rates of evolutionary change for these traits, so sperm length and egg length and width. And you can see that I've highlighted for you the diptera. You can see the red circles mean faster evolutionary rates, and the blue circles mean slower evolutionary rates. And egg length in the diptera, surprise, surprise, Drosophila, much faster than on average. So, but the Drosophila, not all the diptera, you can see that there's only some red dots, not all of them are red, whilst the egg length is actually slower. And in contrast, the Lepidoptera are doing something opposite to that, not all of them again, some clades are, but not all. Sperm length is evolving slower than egg length, which is evolving slightly faster. Again, looking at sperm, leg, and egg dimensions, narrow and long eggs are positively correlated with longer sperm. So you have narrow and longer eggs fit longer sperm. And in contrast to the right-hand side, squat eggs, which are more typical of Lepidoptera, so short and wide, also correlate positively with longer sperm. Why is that? That's a big question. Could it be morphological? Could it be, does a longer sperm need a larger egg to be accommodated? Is in all insect species that the whole of the tail goes inside the egg? Not always. In some species it does, but not always. Could it be pleiotropy? Could it be functional? Is there material in the tail that is needed for the evolution of the egg? Sorry, not evolution, what I was talking about. For the activation, I meant. And here's again that diptera type of pattern, where you can see that I've highlighted in red on the left-hand side, actually Drosophila, which does the Drosophilidae, they do something different from the rest of the diptera, whilst in Hymenoptera is a more homogeneous type of pattern between sperm length and egg length. And just kind of the last few points to conclude. So I mentioned to you that the micropyle is not just a pore. This is an amazing diagram of what you look like when you're cutting through the chorion on the top of an egg. So you can, this is not mine. I've acknowledged the author. I've taken it from the literature. So if you go back to that image I've shown you of the butterfly egg, so this kind of rosette pattern, this is something that it's during development occurs, the kind of cells that imprint during the development form that pattern. And the pore is in the middle there. You can see these little dots, and you can see that the canals don't go through in a straight way. That's another super cool thing about the canals of the micropiles. Sometimes they do U-bends. Sometimes they go diagonally. Sometimes they go into this sigmoidal shape. It's super cool. I don't think there's anybody looking at why they don't go straight into the egg. Why would you go into take a detour if you're trying to go and fertilize the nucleus? I have no idea. And finally, what we found here was that, so we looked inside of the canal itself, and we looked that actually there's a positive relationship between the width of the sperm and the micropore width, but the pore is about 3.5 times wider than the sperm. That is really interesting, because that means there could be enough space to move. I think that's a big unanswered question. How is the sperm entering the egg? And I think that looking at this morphology can help us. Oh my gosh, I'm going over. I want to just very quickly go to the conclusions, which is yes, these sperm, egg, and length traits coevolve, and that longer sperm, females face a trade-off. Longer sperm is associated with longer and wider eggs, and the micropyle pores 3 times wider than the diameter, and therefore this could leave space for channeling and actually helping the sperm enter. Thank you very much for listening to me. Hi.
Thanks, great talk. I wonder if you have the power to look at the micropilot traits to see if they evolve as fast as the sperms. Like overall, the eggs might be slow, but like the FRT, the female reproductive tract, evolves quickly in some species.
So can I make sure I understand the question? So is the micropyle pore, rather than just looking at egg traits, looking at the actual pore and canal? Is that the question?
Yeah, if that evolves, if you look at the evolutionary rates of the micropilot traits,
That is a really good question. The work I did before, I was actually looking at whether micropiles could have a secondary function, because when they are laid in the outside environment, the eggs, could the micropiles still help with oxygen, for example, exchange? This passive exchange between water and oxygen. I found that there was a positive correlation between if an egg had more aeropiles, which are for oxygen exchange, they also tended to have more micropiles. So perhaps that trade-off is really strong in the eggs, and therefore the kind of female has to work out whether to invest more in having more micropiles or aeropiles to let the egg survive once it's laid. But I don't know whether I've answered, but I'm going to catch you in the coffee break. Yes. Do we have time for that? Yeah, 20 seconds.
Do you think that both the micropiles into the egg maybe have something to do with female choice, with selection of sperm maybe for better swimmers in a more hostile environment?
The question was a really good question, which is about what is that canal doing? Could it have a selection? So could it be cryptic female choice? And I'm going to put a grant in to see whether that is the case or not. Thank you very much.
All right, sorry about that, everyone. Just needed to find the talk. So, hi, I'm Kayla, and today I'm going to talk about the consequences of evolving under selection from multiple mutualistic partners. So, mutualistic interactions are absolutely vital for life. Species depend on one another to fulfill an array of essential fitness functions. And I think one of the most pressing questions in the field of mutualism today is how two species with potentially conflicting fitness interests maintain long-term cooperation when these interactions are also characterized by genetic diversity. And so we predict that the maintenance of cooperation in these interactions depends on there being opportunities for repeated interactions between specific partner lineages across generations. And when this occurs, these partners can exert strong selection on one another for the maintenance of beneficial traits and the inhibition of exploitative traits. Through time, this can also lead to increased specializations as these partners co-evolve with one another, and perhaps even derive greater rewards through the interaction as a consequence of specialization. And so here I depict this a bit here, and you see these leaf beetles and these dots represent symbionts, and these are two populations of mutualistically interacting species. And at the beginning of this interaction, we might observe genetic diversity, but when there's opportunities for coevolution or reciprocal selection across generations, we then would expect to observe a reduction in genetic diversity as beneficial partner genotypes are favored by selection, and there's an increase in specialization, and long-term cooperation is maintained. But this is often not what we see, and in fact, genetic diversity is actually quite rampant within mutualistic interactions. We observe instability and partner switching, and we don't really understand how these promiscuous dynamics arise and are maintained within these mutualistic interactions long term. And so, what are the consequences of evolving under selection from multiple mutualistic partners? And this is a question that I address using experimental evolution of mutualistic yeast. So experimental evolution is a really powerful tool to address these types of outstanding questions in evolutionary biology, where examining natural relationships can be really messy. So we can break this down to directly test this question. And I'm using two yeast strains that are reproductively isolated from one another, making them a decent model of species interactions. One strain has been engineered so that it's oxytrophic for leucine and overproduces tryptophan, and the other is oxytrophic for tryptophan and overproduces leucine. So by growing these strains in media that's lacking these amino acids, I can select for mutualistic interaction. And I did this using several experimental treatments. In the high partner fidelity treatment, strains were co-passaged one another each round of selection so that they saw the same partner each round of selection. And this occurred for 54 rounds of selection. So in this treatment, there were opportunities for specialization between partners and also potentially pairwise coevolution. In the low partner fidelity treatment, strains were co-passaged with one another, but throughout the course of experimental evolution, partner switching occurred. So in passage 0, for example, partner A was paired with a partner B1. After 3 rounds of selection, partner A continued to evolve, but we introduced a new partner B2. After an additional 3 rounds of selection, partner A continued to evolve, but introduced a new partner B3. And then finally, after another 3 rounds of selection, we continued to evolve partner A and reintroduced that original partner B1 from the 3rd round of selection. We continued to rotate through these 3 mutualistic partners so that there was co-passaging and interaction between partner A and the rotating partners for a total of 54 rounds of selection. And this treatment was carried out 2 directions so that both partner A and partner B were the continually evolved mutualistic partner. So in this treatment, there were opportunities for there to be selection between these interacting lineages. We expect the efficacy of selection in each pairwise combination would be lower than that in the high partner fidelity treatment. This creates opportunities for generalism and then potentially multi-partner coevolution. Each of these treatments was independently replicated 5 times. And they also included controls. And so here, partner A and partner B were singly passaged without contact with one another, and they were evolved in media containing all the amino acids for 54 passages. And then all of these strains were derived from the same isogenic ancestors. All right. And this work was completed with the help of UMLAC. At the end of 54 passages, strains were paired with one another and plated on media that was lacking amino acids, and I measured the growth of the 2-dimensional growth area of these strains over a 4-day growth cycle. All right, and I'm gonna begin by showing the results of the high partner fidelity treatment. So here on the y-axis, I'm showing the full change in growth area relative to the ancestor. So each of these data points has been normalized to the value of the ancestor across each of the 4 days. In green is the control, and in orange are the high partner fidelity strains. And you can see that evolving under constant selection from a single partner with high partner fidelity results in an increase in growth relative to the control. So we see an increase in mutualistic rewards or in benefits within this interaction. But we don't see this outcome for these low partner fidelity strains. So this is the same axes for this plot. Again, in green, I'm showing the control. In yellow, I'm showing the low partner fidelity A strain. So here, this is where A continually evolved with a new partner B. And in then in blue, I'm showing the low partner fidelity B strain here, where B continually evolved with the rotating partner A. And there's no growth benefit in these multi-partner selection treatments relative to the control. And this is especially impactful when we consider that the strains that are being paired with one another in these low partner fidelity treatments share an evolutionary history with one another in this selective environment. These control strains are being paired for these assays, but were evolved separately from one another, sharing no evolutionary history. So truly, this multi-partner selection treatments, they really constrain the evolution of these mutualistic benefits. And then finally, I'll compare the high partner fidelity treatment to the two low partner fidelity treatments. And we observe that evolving under high partner fidelity yields an advantage relative to evolving with multiple partners. And this growth advantage occurs very early in the growth cycle of these strains. So this advantage is seen as early as day 1, and then persists throughout much of the growth cycle of these strains. And this is really important because when there's competition in nature, for example, having these early advantages is going to give these strains a great advantage relative to other strains where there maybe is a lag in the growth. All right, so here I'm showing this data just in a slightly different way as growth curves. So on the y-axis now is the fold change in growth area. And the next several slides, this is how these plots will look. Again, we see the same outcome, that all of our experimentally evolved treatments exhibit increased growth relative to the ancestor, with high partner fidelity yielding the highest outcomes. And so next I wanted to examine whether or not selection was actually operating in these interactions. So to do this, I paired the evolved strains with either their co-passage partner or with an ancestral partner. And so here I'm showing the data for the high partner fidelity treatment. And the solid line is when high partner fidelity strains were paired with a co-passage partner, and the dashed line is when they were paired with an ancestor. And you can see that being paired with a co-passage strain results in a significant increase in growth relative to being paired with an ancestor, providing good evidence that selection is actually operating on this mutualistic interaction, and mutualistic rewards are increasing as a consequence of the interaction, rather than, for example, one strain being better at growing regardless of who its partner is and just being able to generally derive better benefit and support the growth of the strains. And we see the same outcome for the low partner fidelity A treatment. When strain A that's continually passaged is paired with its co-passage partner B, its growth is higher than when it's paired with the ancestral partner B. Again, providing evidence that there is selection acting on this interaction within this mutualism. But we don't see this outcome for low partner fidelity B. So when the B strain that continually evolves is paired with its co-passage partner A, it gets no fitness advantage relative to being paired with the ancestor, indicating that selection in this interaction is likely quite weak. Or we're not seeing any increase in mutualistic benefit. So what are the consequences of evolving under selection for multiple mutualistic partners? First, we do see that it evolutionarily constrains mutualism. We only observe an increase in fitness benefits under that high partner fidelity treatment relative to the control. But we also see that mutualistic rewards and the strength of selection varies quite broadly within these low partner, multi-partner selection treatments. In one treatment, there's pretty good evidence that selection is operating in that interaction, and mutualistic rewards can increase. There's potential there for that relationship to change. In another treatment, we see really no evidence that there's strong selection operating in that interaction. And I don't show this, but in our treatment where we also see the selection occurring, the effect of the interaction varies broadly across our replicate lineages as well. All right. So in the future, I'm going to continue to explore this system and test whether or not there actually is ongoing coevolution across these treatments, and what the ecological consequences of these relationships are. Examine the consequences of long-term multi-partner coevolution. Given more evolutionary time, would we observe an increase in mutualistic benefit in these treatments where selection is acting? And also evaluate the relative consequences of multi-partner coevolution versus one-sided evolution. Just a quick plug, I'm starting a lab at Florida State in January, and my lab will continue to use experimental evolution and pair that with evaluation of natural insect symbiont systems to address questions related to the causes and consequences of genetic diversity and mutualism, and considering community-level coevolutionary dynamics. And so with that, I'd like to thank my lab and take questions. I started late, so I don't know how long we have, but thanks. I saw this.
Really fascinating stuff. What distinguishes the different strains?
Sorry, what do you mean?
Like in your low fidelity, like you said, you have partner A, partner B.
Oh, yeah.
You identify that some are leucine and others and so on and so forth. But there must be other different, like how different are they?
So they are all derived from like isogenic ancestors. So it's just that we're starting and stopping so they all start out initially the same. And then we're allowing de novo mutation to arise in these lineages as they see each other through time. And they would diverge from one another, we expect, over the course of experimental evolution through opportunities for selection. Okay.
Are they intractable genetic bottlenecks? Genetic bottlenecks?
I don't think that we're transferring really large quantities of cells across passages, so I don't think that there's going to be a large impact of genetic bottlenecks. We do reset the population to an OD600 reading of 1 to normalize the cell density so that the partner distributions are equal across the two strains.